Or expectations. On a number of levels, which I find very and why did I want to raise that's a challenge. Wow, they just sold out your building.
It's not unusual, we're used to it, but I studied in the second plane, second fiddle. No, it's a color.
Oh, it's all within.
No,
But you're cohabiting.
still a very significant building. So they are.
Howdy, everybody. Oh, it's 11:30 and we sound like we just woke up. Howdy. Howdy. Okay. My name is Doyla Wilson. I am introducing Dr. Choudhury. She is the assistant professor in the Department of Pharmaceutical Sciences at Texas A&M Health Science Center College of Pharmacy, Kingsville, Texas. She also serves as an adjunct assistant professor in the Nutrition and Food Science Department here at Texas A&M College Station. She received her bachelor's in zoology, botany, chemistry, and her master's in molecular biology, biophysics, and genetics, both at the University of Calcutta, India. She received her PhD in medical pharmacology at the University of Missouri, Columbia. She has achieved the One Health Initiative Award, the American Diabetes Association Mentor-Based Postdoctoral Fellowship, the Bill and Melinda Gates Grant Challenge Award twice, and over 20 more different honors and awards. She currently serves as a reviewer in the Diabetes Care, a member of 5 professional affiliations. Her research interests include prediction of disease risk, genes and the environment, and pathology. Her topic today is the role of epigenetic regulator, Sirt3, in diabesity, a contemporary view. I present to you Dr. Choudhury.
Thank you for a nice introduction, and thank you for the invitation to present my research interest here. And today I'm going to talk about one interesting molecule, and while I was putting the slides, I thought, as this is a graduate student seminar, rather than putting all science data, why not put together a nice story, how I faced the challenge, how I overcome that, and how I actually become an independent faculty with the science. So the project basically started from an accidental data, and it actually, we found a nice molecule which might have a role in diabetes, obesity, and other metabolic syndrome in the long run, and we might find a therapeutic target in that molecule. So The title of the topic is Role of an Epigenetic Regulator Sirt3 in Diabesity, a contemporary view. So the epigenetics, which is becoming more and more interested now, and why I'm going to talk about more detail in next few minutes. But first, I'd like to divide the topic in a few points. And first, I would like to describe some of the obesity and its complications. You might all know, but I just want to refresh your memory. And then I'd like to talk about the Sirt twins, which is the most important topic for today, and what we found in high-fat diet-induced obesity and the Sirt dysfunction, and the conclusion at the end, what we can actually do in the future. So obesity, what is obesity? You all know that, but this is actually a true picture of obesity. It's a heterogeneous complex disorder, and it has multiple etiologies. For example, this guy is just a 40 years old, 5 feet 6 inch, 250-pound weight, and his BMI was 40.3. And you can see like everywhere he has extra fat. And he not only is obese, he has problem with several other organs. For example, he has hypertension. He was insulin resistant. He has a joint pain in almost every joint. He had problem with his eye, and most probably he will be blind in 20 years. So it's a devastating disorder, and it can regulate like not only the fat, it actually can relate to other organs in your body. So the identifying a molecular mechanism which might be underlying all this problem is the mission of this our modern medicine, but it's not easy to find one molecule, okay? So it might be like you have to find a regulator or an important regulator that might have several effects and which we can actually target and may able to reverse it. So another point on this obesity topic is the childhood obesity, which is becoming scary nowadays. 1 in 6 US youth is obese, and they are the future of our country. And so this is becoming scary, and we have to do something. Otherwise, this might lead to a future which everybody are somehow affected of this epidemic. So these are the medical complications of obesity. As you can see that every organ of the body is affected. Heart is affected, lung is affected, you have bones, they are affected too, and reproductive organs are affected. But I am interested in fatty liver disease, in the liver. And liver is one of the important organ in your body. And I worked on alcoholic fatty liver disease in graduate studies, but in my postdoc, I started working on non-alcoholic fatty liver disease. So I have the both background of fatty liver disease. And what I understood from the main point of this fatty liver disease is you have a liver which are tons of fats there. You have inflammations comes along, it becomes NASH, which is non-alcoholic steatohepatitis, and then it actually leads to the cast tissue, which replaces the liver cells, and it can lead to the liver cancer. And so you don't want to go that further. So what I am interested, and many people are interested to find it, we want to stuck here. And we have to find it and reverse this fatty liver to a normal liver so that we don't go forward with the next step. So the target is basically finding a molecular mechanism here so that we can actually reverse the disease as early as possible. Don't go so further alone. So this is the first data of this project started, and it was really accidental. So what happens that Dr. Johnson, she was a trained by physicists, and she was doing some kind of mass spec optimization, and she needed some samples. So what I asked her, I have some samples which are liver samples, and we put the mice on normal diet for 16 weeks, high-fat diet for 16 weeks, and I want to see that if they have a acetylation in those liver proteins or not. But I'm more interested in histone proteins. So she said, I have never done it, but I can do it for you because I need to optimize my new mass spec, so I need samples. So I said, okay, I'm going to give you the samples. So she actually went ahead and ran the 1D electrophoresis. And there's one student, she was doing her master's in chemistry, and she wanted to actually help the project and get a credit for that. So it was a nice combination of, you can say, physicist, chemist, and a biologist. I wanted to have the data. So, and funny thing is, she was interested to do PhD in chemistry, but she is now a second-year biomolecular science PhD student. She had got the interest in biology, and she basically changed her degree to biomolecular science. So what we found that, if you can see here, these are the high-fat diet liver, and we found tons of band here. So these are basically hyperacetylated band here compared to chow-fed diet. So we got interested and we thought, so what's going on? So let's take up that. But we needed a money dollar to do the project. So we applied for this NORC grant and we got a NORC pilot funding to look for this project, what's going on in here. So you know this picture very well. So I was interested in epigenetics and especially the acetylation. I was interested in histone acetylation because histone is one of the main topic in epigenetics. But we didn't get actually histone proteins in that picture. We got all the proteins non-histone. So I looked into the publications and I found that suddenly there's tons of paper coming out on acetylation and the importance of acetylation in biological pathways. So it's basically a new phosphorylation because we always like to think that, okay, if you have a protein molecule, if you have a phosphorylation, that might activate the whole pathway. But it seems like in 2010, almost like a 400 papers came out. I just today typed the acetylation word, and 2014, 1,700 paper came just this year on acetylation and role in different kind of biological pathways. So this is really become a new area of research and which might actually lead to a huge interesting pathway. So what is this acetylation really means? So you have a protein, it can be deacetylated or acetylated depending on two sets of enzymes. It's called histone acetyltransferase and histone deacetylase, okay? So if your protein is going to acetylate it, your HAT activity or the expression will go up. So it's kind of like a two-way experiment. So if this HAT goes up, you have more protein acetylated. You have HDAC goes down, then also you have more protein acetylated. But if you have HDAC goes up, then you got the protein deacetylation. So it's basically a combination of a balance of HAT and HDAC. And what is Sirt3? Sirt3 is basically one type of HDAC. So what Sirt3 really means? It's a silent information regulator. That's the full form of Sirt3. And the gene is responsible for different kind of cellular regulation in yeast. And it was found in yeast first. And it has the activity which removed the acetyl groups from lysine residues within the proteins in the presence of NAD. So it needs NAD to work on. So it's a NAD-dependent HDAC. So this is the one class which are dependent on NAD, and it's called class 3 histone deacetylase. And it's classified as NAD plus dependent deacetylase. So where we found this, we found in brain, heart, skeletal muscle, liver, almost everywhere, depending on where the more mitochondria are, they are more expressed in that tissue. And when we started working on it, mostly it was studied in cancer or neurodegenerative diseases. But then when we started, and within a year, actually more metabolism paper came out, and more research has been many people actually working on this aspect. So there are 7 types of Sirt2s are there, and what we are interested in Sirt3. So this can be located in the mitochondria, nuclear, or cytoplasm, but certainly are mostly in mitochondria. Why I am saying mostly? Because there are few papers actually are saying that it may be in the nuclear, but maybe there is a indication of long form or short form, but many papers actually showed specifically that Sirt3 is in mitochondria. So there are 3 mitochondrial Sirt3, Sirt3, Sirt4, and Sirt5. And as you can see that Sirt1 are mostly studied because you can see more substrate are found on Sirt1. But Sirt3 has not been studied that much. So this is a nice picture where basically you're showing that Sirt3, Sirt4, and Sirt5 are mitochondrial, and they basically take part in energy regulation. So mitochondria are the production of energy production organs. So they actually take part in energy regulation. So when we started, we hypothesized that we looked into publications, and it seems like caloric restriction caused the increase in Sirt1, which can deacetylase many protein, and that leading to the longevity, which is the aging. So, and there are huge interest in that because everybody wants to live longer, and so they are more interested in how can I get more Sirt1 in my body. So what we thought, maybe in our case, we are thinking about high-fat diet, so it might just goes opposite. So if you have a high-fat diet, your Sirt1 will go down, and more hyperacetylation happens, and it can actually have some effect on nucleus or mitochondria, and it can lead to fatty liver disease or any other more complications. So we mostly started the thinking that Sirt1 is our molecule, which actually at the end didn't happen, but we thought Sirt1 is going to go down under high-fat diet condition. So that's another lesson for us, that don't be so biased that everything will not be 2 + 2 in science. So that's an important point in science. So our question was, does a high-fat diet modulate the Sirt activity to impact the acetylation of proteins or the pathways involved in obesity and comorbidities in liver? So main question was, does a high-fat diet affect Sirt1 or not? So this is a simple hypothesis we did. High-fat diet, it will cause modulation of Sirt expression, but we also took actually account for the HAT, because we thought maybe HAT also be modulated, as I showed you before that HAT is another protein which are involved in acetylation. So next step is modulation of more protein acetylation, and that can lead to the obesity and obesity-linked diseases in liver. So this was a general hypothesis. So then what you need to do, we need to investigate at cellular and molecular level, and we have to do work in the mouse. Even though this was a joke, but we have to start somewhere to actually study the molecular mechanism. So this was our experimental approach, very simple approach. What we did actually, we have a control mice and high-fat diet mice, and we have 45% fat. We put it on 16 weeks. We did all the general study, like a general hallmark of obesity, food intake, weight gain, everything was as usual. Like it got actually fatty liver at the end of 16 weeks. So we took the liver out and we had like a separated into different experimental aspect. We did the mass spectroscopy. We did different molecular biology techniques. So again, this was the first picture I showed you before, that high-fat diet and chow, and we did like a 4 or 5 times just to make sure the first time was not fluke, and we got the same result again and again. So it's, as you can see that high-fat diet is causing more hyperacetylation than the chow-fat diet. We got almost 139 proteins which are hyperacetylate. So we got this protein, but we didn't actually look carefully what are the proteins are. We just got interested. I didn't get any histone protein, so I just, okay, let's see what's the next step. But I didn't look carefully. And that's why we actually ended up working too much, because we didn't look carefully on that list. So I'm going to talk about that in a few seconds. So here the basically mass spec data, it's showing like a more acetylation or less acetylation depending on several spectra, but the bottom line is more hyperacetylations occur under high-fat diet condition. Then we looked into the molecules and we found that most of these molecules are related to metabolism. Somehow it's related to metabolism, glycolysis, amino acid metabolism, ketone body synthesis, stress metabolism. So all are related to some kind of metabolism, like metabolism pathways. So then we started looking at few of the proteins which we got interested, because we can't do 139 proteins. So we thought, let's do few, and I'm just showing few of them which are actually involved in metabolism. And as you can see that more acetylation happens in case of high-fat diet. So this was a Western blot. Actually, you cannot buy the acetylated antibody. So what you have to do, basically, you have to immunoprecipitate the lysine antibody. Then you have to run whatever protein your interest is. So we did the immunoprecipitation, we run the interested protein, and as you can see that, yes, under high-fat diet conditions, all are hyperacetylated, okay? So now we are happy that whatever our mass spec data are showing, we're getting the same result in individual Western blot. So the next question came again. So protein are more acetylated. So what should I do? We should look into what's responsible for this protein acetylation. So we looked into HAT, we looked into HDAX, and we looked into Sirt3. So here some of the Sirt3 Western blot, and as you can see that Sirt1, Sirt3, Sirt4, I didn't put the Sirt5, but we already did it. Nothing actually changed. The protein expression of Sirt2s did not change under high-fat diet conditions. So later, actually, after we published it, our collaborators showed that they got actually Sirt3 decrease under high-fat diet condition, but they put the mice on 60% high-fat diet on 12 weeks. So maybe if we keep these mice for longer time, we might get that Sirt3 gene lower Sirt3 gene expression or the protein expression. But in 12 weeks, we did not get it. So what we thought, okay, maybe the histone acetyl transferase is responsible. So we did the HAT activity assay, and HAT didn't change at all. So here is HAT. So HAT activity, you can see that it didn't change, and the activity kit was available, so we actually were able to do it right away. But there are no kits were available for actually measuring this Sirt1 or Sirt3 or any kind of Sirt2 activity. So whatever the kits available, those are basically for drug screening. So we talked to the company and they said, we never used it for any cell culture stuff, we just screened the drug. But then I talked to the technical support and kind of like optimize, even though it's only 3 grafts, it took like 6 months to do it because we had to optimize. And this is the first time we showed that you can use the commercial kit, but do the Sirt1 and Sirt3 activity in cell culture, like in an extract, not the cell culture, in a protein extract. So what we found that we did the Sirt1 activity first, and we didn't find any change in Sirt1. Remember that when I was showing the hypothesis, I was telling that, okay, Sirt1 will go down. That's basically our hypothesis. And we didn't find it, and we got very frustrated. What's going on? We're not getting HAT activity and no change. We're not getting Sirt1 activity. So we kind of stopped there, and we didn't even think about Sirt3. And then we actually looked back to that mass spec list, and we found out that almost 80 to 90% of that list were mitochondrial protein. We missed that in the first place. We didn't look into carefully, but we looked at that list carefully, and we found that almost all proteins are mitochondrial. And Sirt1 is not mitochondrial. So our whole hypothesis was wrong in that. So we looked into then mitochondrial activity, and which is Sirt3, and we found that Sirt4, Sirt5 did not change, but Sirt3 activity actually went down. So that time we really was like very happy to see that, okay, even though our hypothesis didn't work what we wanted, but there is a reason why we are seeing so much hyperacetylation in high-fat diet-induced liver. So Sirt3 activity going down. So the next question was what we can find in the literature about Sirt3. So what we saw, all the papers are related to Sirt3 are under dietary restriction. And they're showing that when Sirt3 increased, they have different kind of pathways which actually at the end adapt the mitochondrial adaptation, and this is good. But in our case, we're talking about high-fat diet. So basically, we're talking about the reverse thing. So this is the paper actually came out recently, and almost like a similar time, and most of the from the collaborators are our known people who actually shown that yes, Sirt3 actually involved in metabolism. So in 2007, Wording group actually shows that Sirt3 is in the mitochondria, and it's mostly in brain, heart, liver, and brown adipose tissue. And it's in the mitochondria, it's not in the membrane, it's not in the cytoplasm, and not even in the nucleus. So Sirt3 is strictly mitochondria, so already they showed that, and we actually got the same result that mitochondrial protein are hyperacetylated. Another paper, and the same thing, they are showing that, and this is very interesting that they have these Sirt3 knockout mice which are viable and which are metabolically unremarkable. So this is the liver histopathology where they didn't find any changes, okay? They looked into mitochondrial biogenesis and different complex actin, complex protein. They did not find any difference in knockout and the wild type. They looked into body weight, body fat. They looked into bone mineral density, bone mineral content. They looked into respiration. They looked into food intake. There's no change between knockout and the wild type mice. So what they actually concluded that maybe under normal condition, nothing is going to happen. But if you are putting under stress or something, it might change. So in 2010, just before we published, they showed that in fasted condition, fed condition, wild type knockout, nothing happened. Fasted condition, ATP production went down. There's a defect in fatty acid oxidation. They have a cold intolerance. They also are hypoglycemic. So there is some changes under stress condition. So Then the next thing we wanted to find out that, okay, if the Sirt3 activity is changing, and as we know that Sirt3 depend on NAD because it's a NAD dependent HDAC, we wanted to look into NAD concentration. And as we looked into the NAD concentration, we found that under high-fat diet condition, NAD goes down, and it's actually inversely proportional to the change of body weight of the mouse. So it's a nice data which is supporting that Sirt3 activity is going down because of NAD concentration also was going down. Then we looked into the oxidation of those liver samples, and we looked into that, and we saw that there are more protein oxidation happening in the liver under high-fat diet condition. So high-fat diet increases the mitochondrial protein oxidation in liver. So the next thing is, we are like, okay, everything is happening, but is the Sirt3 is the real mediator? How can I prove that? Because maybe it's happening, maybe it's not a direct relation, maybe it's an indirect relation. So what we did actually, we needed to do the work in Sirt3 knockout mice to prove that we are actually correct. So do you want to make the Sirt3 knockout mice? It will take another 2 years. So what we thought, okay, let's just go ahead and do the collaboration with some of the people who has Sirt3 knockout mice. So started the collaboration with Dr. Geis and Dr. Sapp, and they actually was wrapping out their Sirt3 knockout mice project. So whatever this liver sample they sent me, it was gone within 2 days. I didn't get any data. So at the end, I went to Dr. Khan, who has tons of liver sample, which was on the high-fat diet. So we looked into the next thing, which is basically Sirt3 knockout mice liver, and we actually ran the same samples, which are those acetylated samples, and we saw that same results from the one we got it under high-fat diet. And the protein expression did not change, which means the protein is not changing, only the acetylation level is changing. So the next thing we have, okay, now we have to revisit our hypothesis that this is not Sirt1, Sirt3 is going down under high-fat diet condition. So next thing we wanted to check, what's going to happen with all these electron transport chain complexes? Is there any effect on electron transport chain complexes? So now we have a mice, knockout mice, which were on the high-fat diet, but again, these are high-fat diet on 60% for 12 weeks. So we saw that under controlled diet, Sirt3 knockout mice did not show any changes in mitochondrial complex activity. So the wording group was correct. If you don't have any stress or anything, nothing is going to happen. But when you put them on the high-fat diet, actually the complex, all the complexes almost actually went down. And these are the expression data. But when we did the activity assay, we saw that all the complex activity actually went down, but this three showed these are like almost significant, not significant enough, but all the mitochondrial complex activity went down under Sirt3 knockout mice under high-fat diet condition. So we are really happy to see that what we thought, it's the same thing, that high-fat diet can actually regulate Sirt3 in the liver. So the conclusion was high-fat diet feeds liver showed protein hyperacetylation. It's associated with decreased Sirt3 activity, at least in our case, correlated with the decreased NAD+. It increased the oxidation of mitochondrial protein. The mitochondrial respiratory chain complexes dysfunction happens. So this is a simple conclusion. So, but the good thing happened after we published the paper, and when we published the paper, we want other people to read the paper. So this is actually after 2 years, we published the paper in Biochemical Journal, and they actually send this acknowledgement that your paper was most read paper in our 3 years of history, and which is very like a accomplishing for us that, okay, somebody at least read the paper and it got cited tons of time. So now the huge group, the Khan and Bardeen, both are working on this, and they are showing that Sirt3 knockout actually can lead to accelerated obesity, insulin resistance, inflammation, NASH, steatohepatitis, different metabolic syndromes. So Sirt3 is an important regulator, but these are all basically just started. These are all like a tip of the iceberg. So we have to work more and more to find out what's going on. Can we actually make something out of this? So the next goal, basically, these are like a future plan, and we have to think more and more how we can actually increase the Sirt3 if in case Sirt3 is the one regulating this metabolism. So we're thinking about different kind of adenoviral delivery in mice experiment or different dietary supplement which might actually increase Sirt3, and we have just started working on one nice molecule PQQ, which you can find actually in CVS pharmacy or Walgreens, and they actually are found in some kind of fungus in Japan, and they eat that actually. So that looks like a potential Sirt3 activator. So we are working on that, and the company Mitsubishi, they're basically making those PQQ. It's called BioPQQ, and we are planning to do more study on that, and we're also looking at some other drugs, and most probably in future like a clinical trial on that. So at the end, I'd like to thank you, my labs, and my postdoc mentor, Dr. Jacob Friedman, and all the other collaborators, and the funding which whole Sirt3 study was started by American Diabetes Association funding and the NORC pilot funding. Any question I'd like, I'm happy to talk. Thank you.
So is this unique to obesity? Because you compared, obviously, groups of animals that were fed a high-fat diet with specific composition generated by a company using semi-purified ingredients to a chow diet, which of course is extremely heterogeneous and difficult to define.
Right.
So is there something, is it just basically body mass that's causing this problem, or is there something unique about the composition of the high-fat diet that you think drives this phenotype?
Right. So that's basically my next question, and we are actually looking at the like if the we are talking with the research diets actually having the purified diet, if there is anything, because this diet comes up with several things. We don't know which components are those, and is it like a really obesity effect or not. But what the other like a project which was on the human actually. So they basically had like a high fat, not a high fat diet, obese people, and like a healthy control people, and they have seen like a less activity in Sirt3 samples, like some of the biopsy samples. So it might not be just our mice study, like a research diet study, but it might be in general in human also. No, we actually didn't do that part. So that's basically the next start. Can you actually recover it and reverse the whole thing? It might not be like our idea. It might, you cannot reverse like 100%, but at least like maybe like a back into some normal activity.
Do you have the mitochondrial number?
No, we didn't look into the mitochondrial copy number or anything, but only the mitochondrial activity.
Is it possible that Sirt3 activity decreased is just because the last mitochondria?
Yes, we didn't look into that, but as I showed you that the data, like the TFAM, didn't change in the knockout mice at least.
So can you speculate what is driving down the steady state levels of NAD that you feel is primarily responsible for the reduction in the Sirt3 activity?
So that's a good question. Like, is it like a diet or something, or we are thinking that because NAD can come from the dietary source, like a normal dietary source, and if there any other like a gene or something, for example, NMN, which might be another like which actually help in producing NAD, like if there any problem with gene or anything, so that might another probability. But there's another thing which they found in human. There is a mutation in Sirt3 in obese people. So there's like a genetic polymorphism they found in Sirt3. So it might not be only NAD, because as you can see that it's not like really like a gone or anything. NAD is there, still there. So so there might be several factors involved.
And is the transport of the acetyl-CoA levels, in other words, the substrate that you're acetylating all these proteins with, is that unchanged?
We didn't check that, but some other papers show that it's actually acetylated.
Sorry, what's the cell?
Acetyl-CoA synthesis.
Oh,
Yes.
any other questions from our distinguished audience?
What was the last? I missed the last one. You said the last finding that you had was it decreases Sirt3. It's a microprotein, and then you said it releases faster, and does what else? I only got you said that the conclusion had good parts, and I only got the first two. To your paper, when you wrote the paper, just before you wrote the paper, you said that you had come up with a conclusion, and I only got the first two. What are the last two?
So the complex activity you are talking about?
Yeah, just before you wrote the paper. That's why that says just before you wrote the paper. That one. Yeah.
Leave that slide up.
Okay.
Thank you.
Any other questions? Okay, then let's have a big hand then for Dr. Choudhury. Remember, she'll be meeting with students in the faculty lounge for lunch. There'll be plenty of time to ask challenging questions and acetylate yourself by eating too much. Or is that acetylate yourself too little? Whatever.
Okay, sir, it's fine.
Yeah. I'll send you that email then. Thanks, Dave.
Okay, I took it out. That's fine. Okay.
Very It's very provocative, and it's probably happening in other tissues as well, and
Yes.
we'll probably explore that.
Yes,
Find
yes.
out wonderful things.
Yes, especially like I'm interested in the adipose tissue. What's going on, and because brown adipose has more mitochondria. So, but I wanted to talk to you. So I'm going to start one project on hyperdiet, sucrose. I wanted to make sure before I start the project, like I'm doing that correctly.